All right. I think I'll go ahead and kick things off, and people can continue to filter on in as they do. But yeah, thanks so much, everyone, for joining us. Good crowd. Hopefully, you guys are all having as nice of a day as I'm having here. Really excited today. Today, we're being joined by Dr. Peter Agre. Dr. Agre is a medical scientist and 2003 Nobel laureate in chemistry. Dr. Agre studied chemistry at Augsburg College and received his degree in 1970 and went on to study medicine at Johns Hopkins in 1974. Dr. Agre later returned to the Johns Hopkins School of Medicine faculty, to join the faculty in 1984, where he rose to the rank of Professor of Biological Chemistry and Professor of Medicine. He's currently Bloomberg Distinguished Professor at the school, and since 2008 has been Director of the Hopkins Malaria Research Institute at the Bloomberg School of Public Health. He's best known for his discovery of aquaporin water channels, for which he was awarded the Nobel Prize in 2003. He now focuses his attention on combating malaria in his laboratory and in the field, where he oversees efforts in Zambia, Zimbabwe, and the Democratic Republic of the Congo. Without any further ado, I'll turn it over to Dr. Peter Agre. Thank you so much for joining us.
Connor, I'm pleased to be with you. I just apologize if my voice gets raspy. I have Parkinson's disease, and that sometimes makes it a little raspy. I plan to tell you a little bit about how things worked out for me and put that in the perspective of scouting. I think the lessons learned as a scout are oftentimes important all through one's life. Maybe decades later, you rely upon positions and information and attitudes that you pick up very early. And I feel like I was very fortunate and was born in Minnesota to a family that appreciated scouting. I'm going to put my photographs on screen share. I'm hung up here. Let's try it again.
Yeah, I'll give you some access to that. Here you go. Should be able to work now.
Screen share. There we go. Sorry to be slow. So this is my dad. He was a real chemist. He taught at St. Olaf College and Augsburg College in Minnesota. And he was not a scout himself. He grew up in a tiny hamlet in South Dakota, but always admired scouts and taught chemistry merit badge every Saturday in his laboratory at Augsburg College. And so hundreds of boys in the Twin City area got the chemistry merit badge. Here's my family. Dad, chemistry teacher's bow tie, very strong influence of science and math. And my mom, who was not a college graduate, she was a farm girl, but very religious and very interested in humanities and music. And the complementation of their personalities, I think, was really helpful for my brothers and sisters and me. So I'm the one with the big ears, you recognize on the far left. My brother Jim, who's a year younger than I, and I received our Eagles together. It was autumn of 1965, and my dad carried this snapshot in his wallet for the rest of his life. It was that important to him. Now, part of doing well in life is having an assessment of what your talents and interests are, and we're not all fortunate at the same level. So Jim and I both were interested in science and good at athletics and had a great time in school. And we both became medical doctors, as did our brother Mark, the little boy in the red blazer in the front. My sister Annetta, in the far right in the back, was interested in education, became a teacher, and then raised her family. But my brother Paul, in the light blue sweater on the right, never had those advantages. He was born learning disabled and had a much harder life to live. And Sister Ruth in the red dress in the front was suffering from lifelong emotional disabilities, and it was a challenge. We all grew up in the same family, but we had quite different outcomes. And this is what my mother always, without identifying specifically what we should be doing, but always encourages us to use our talents for the well-being of others. And that is clearly a scout value. I was a soccer player in college. I went to a small college, and because of that, I had little time for other activities. So I studied a lot and played soccer and made my way to Johns Hopkins, where I intended to pursue a career in global health. And using the background as a wilderness camper and scouts, the canoe country trips we take every year made it easy for me to understand that organization is everything if you're traveling. And I spent a gap year traveling in Asia in 1970 on my way to Johns Hopkins, where I studied global health and did clinical training. This is the internship residency group at Case Western Reserve University Hospital, where I spent 3 years, and then started my own lab at Johns Hopkins. And we were originally looking at cholera, which is a horrible diarrheal disease, and then looking at red blood cells as a membrane source of membrane proteins. And I had the great fortune of meeting Bradley Denker, a Hopkins intern and resident, who joined us for a year and discovered a new protein in red cell membranes. That may seem a little esoteric, but it turned out to be a very interesting protein because no one had seen it before. And so I don't know if Brad was a Boy Scout, but certainly the observation that you're taught was very helpful. It turned out this is the protein now known as aquaporin 1. And I had another good fortune of recruiting a technical assistant, Barbara Smith, from a small town in Maryland, who was interested in research in addition to being a medical technologist. And with her careful assistance, we purified the protein in one step. If you look at the polyacrylamide slabs, on the far right, there's a band that's unmistakable. I'll use the pointer. That protein was purified in one step, which is a great opportunity to do something very interesting because it had never been seen before. And with the help of others, it was always with the help of others, we cloned it out and found it to be related to proteins identified in diverse origins, lens of beef cattle, brains of insects, bacteria, and plants. The function was not known. So what can you think of that is shared by all of these different organisms? And it turns out it's not obvious. But part of a career is knowing when to call it quits and make sure you provide time and interest in your family. And I am very fortunate to have met Mary, my wife, back when I was a medical student here at Johns Hopkins 49 years ago. And we have 4 grown children. This is our son, Clark, who was also an Eagle Scout. And so we had this new protein that we were very excited about and didn't know what it was. We talked to lots of people, read lots of papers, and we were stuck. And here's where the work-life balance helped us out. Every summer, we would camp in a national park. The kids loved it. Into Yosemite, Yellowstone, Smoky Mountain. After doing this for a few years, Mary said, let's ask the children which national park they want to go to next year. And the kids all yelled Disney World, which is, of course, not a national park. But we camped in the Everglades. And on the way back to Baltimore, it's a long drive, stopped in Chapel Hill, North Carolina, where I'd spent 3 years as a hematology fellow, and talked to John Parker, a professor there who is a good friend. And it was his insight that put it all together. Membrane protein channel-like, probably, but channels will carry specific solutes like sodium channel, chloride channel. He said that it has been a puzzle in physiology for a century how water gets into cells and out of cells, and some tissues very rapidly. And it was his suggestion that put us on the track of explaining how all membranes have a small degree of water permeability by simple leak through the bilayer, the lipid bilayer of the membrane, but a subset of tissues, red blood cells, secretory glands, and our renal tubules, they have very high water permeability and were believed to have water channels that no one had ever identified them. So this was the idea of another person, which I think was always made obvious as a scout, that patrols do things together. It's very much more valuable to have a team effort. And the notion that if you want to go fast, go alone. If you want to go far, go with the group. And the group was very helpful to us. So we're all familiar with water in nature. It's ubiquitous. Our bodies are 2/3 water. And for the most part, the water is at equilibrium. And this is a tranquil river, the Thelon River in northern Nunavut in Canada, where my son and brother and I paddled. We would go to the Arctic and subarctic every year on our vacation as our kids got older. And it shows that water by itself is not inherently dangerous, but when at disequilibrium, water can be very dangerous. This is the Deaf Rapids of the Seal River at the entry to Hudson Bay. We were paddling as hard as we could to make it through that rapids. So to test the function of this new protein, we teamed up with Bill Gugino, a colleague at Johns Hopkins. We were both young at the time. This was 30 years ago. Bill is now the chairman of physiology at Johns Hopkins, but we were young scientists with a lot of curiosity and ambition. And the test of this new protein was undertaken by expressing complementary RNA for the new protein in the Xenopus laevis oocytes. These are frog eggs. Frogs lay eggs in freshwater ponds. And it's probably happening right now here in Maryland. If the frog eggs are put into distilled water, nothing happens. And this is a control oocyte injected with bufferolone. And here's the test oocyte injected with the complementary RNA for the new protein. So in isotonic salt solution, equal in and out, nothing happens. But if you transfer these to distilled water, the test oocyte swells rapidly and explodes due to the massive water permeability. And this produced great jubilation in the lab by Greg Preston, the postdoc. So a simple experiment like that, which just follows the kind of logic that kids develop and retain, allowed us to make a significant step forward. And we teamed up with many others around the world. That's one of the great things about being part of an organization, whether it's an athletic organization, a scouting program, or universities, to have colleagues that you can rely on and collaborate with. Shown here is Yoshinori Fujiyoshi from Kyoto University in Japan, and on the right is Andreas Engel from the Biocentrum in Basel, Switzerland. And together we solved the structure of this new protein, aquaporin 1, at atomic level. We know exactly what it looks like. On the left, you can see the channel is actually a pore, that like a donut, and in the center, the passage was 3 angstroms in diameter, just big enough for water to cross the membrane. And we teamed up with others to look at where the protein is expressed. This is Søren Nielsen from University of Aarhus in Denmark. Søren was just out of graduate school when we teamed up and solved the location of the aquaporin protein, as we call it, aquaporin 1, in renal tubules. These are the tubules which urine flows after being filtered in our kidneys. And it allows us to concentrate urine when we're fluid deprived, or release dilute urine when we're fluid overloaded. Teaming up with Landon King, shown here, when he was a postdoc. Landon was I don't know if he was a Boy Scout, but he was the quarterback at the Wake Forest football team, and was a natural born leader. And he led the efforts to identify the null phenotype, humans with mutations in the gene encoding aquaporin 1, and studied a family from France. And they look normal, they feel normal, but they are unable to concentrate their urine fully. When we go to bed at night, if we sleep 7 or 8 hours, we concentrate our urine so that we have a relatively small volume of urine in our bladders in the morning. These people have to get up multiple times during the night, and you can imagine in stressful situations that might be very difficult. And there were many aquaporins, a dozen in the mammalian genome, bacteria, mollusks, plants. All organisms have aquaporins, which contribute to the water permeability, and it is a field that really took off because of the collaborative nature. Here's Mark Nepper, former Eagle Scout from Ohio, who directs a program at the NIH, who studied the collecting aquaporin. This is what explains how we can concentrate our urine fully in response to vasopressin, a neurohormone. Ole Petter Ottersen from the University of Oslo contacted us. We collaborated and solved the water permeation in brain, the discovery and analysis of aquaporin 4. His junior colleague, Moham Amiri Moghadam from Iran, demonstrated that the brain damage that occurs after an injury can be ameliorated if we can inhibit the aquaporin 4. Mice that had mutant aquaporin 4 did better. And this has dramatic clinical relevance. Brain injury is all too common, from an athletic injury, a motor vehicle accident, a tumor, a stroke. And this lady had a frontal lobe brain tumor, which grew gradually over many years. It's not a malignant brain tumor, but you can see the size is enormous. And it started to cause symptoms because of the edema. This is an MRI image weighted to the density of water. You can see the tumor is surrounded by all this swelling. The neurosurgeons removed the tumor, and the patient was cured, but it took a long time for the edema to go down. Had that edema increased, it would have caused blindness or permanent brain damage. And this is that lady. That lady is my wife, Mary. She's now perfectly healthy and recovered fully. And so I think the fact that this could happen to us means it's a sort of problem that could happen almost anywhere. Another member of the aquaporin family worked on by Surbhi Raina, a young lady scientist from New Delhi who joined us for a few years and now is a professor at Syracuse University. And there are water channels, the aquaporins, and similar structures, which for me, by glycerol, we call the aquaglyceroporins. And they're present in skin. That many skin creams have glycerol, and that's what makes them very effective. And it turns out that Christian Dior company contacted us a few years ago. They had a natural product which led to increased expression of the aquaglyceroporin theory in skin. And they would, of course, like you to buy this. It's very expensive. I'm not convinced that it's effective, but it shows that the work can be of interest to diverse groups. I showed this photograph. This was on the back cover of one of the French beauty magazines. I showed this to my mother. She was in her 80s at the time and smiled and said, "I think you're doing something useful." That's a nice to hear, but I think some of the other information may be more useful. So I'm going to skip quickly through some of the other biological slides, talking just about the people that did the work. Jen Carberry, a former Clemson undergrad, who did her PhD with us, and she's a professor now down at Duke. She worked on the aquaglycoporins in adipocytes. Those are fat cells that release glycerol when we're fasted. And plants, as I mentioned, have aquaporins. This is a wild type Arabidopsis, a small mustard-like plant. And here's the same gene stock in which they knock down the expression of one of the rooted aquaporins, and the plant compensates by sending out more roots. Each is less efficient. And so this was really all-consuming, but I really felt a hunger to get back to our original interest in diseases of the developing world, global health. And we started to look at malaria. But before we could get very far along the malaria story, I received a call from Stockholm. Connor mentioned Nobel Prize in 2003. You can imagine being in bed at 5:30 in the morning and the phone rings. This is an important telephone call from Stockholm for Professor Agre. Are you Professor Agre? And I was astonished to learn that I received the Nobel Prize in chemistry. My wife called my mother, waking her up in the very early morning, and informed her of this. And mother, true to her farm girl nature, very modest, she said, well, Mary, tell Peter that's very nice, but don't let this go to his head. We're still waiting for him to do something useful. I think many mothers share that. Now, you can't really contain the level of celebration when something like a Nobel happens. I show this with tongue in cheek, and just like to point out that the suggestion that I'm the Wells Liquor best customer is a great exaggeration, but it shows that the public is interested in science and our careers. And the public is the group that supports us with taxes that pay for grants from the National Institutes of Health and National Science Foundation. So here I am with King Carl Gustaf receiving the award, and here I am with my family. And it was wonderful. I have to say, though, that no individual wins a prize like that without tremendous help of collaborators, friends, even competitors. So an individual gets a prize, but the whole field contributes, and we hope this will be of great use to humanity. It still remains to be seen if the aquaporins will be. We had at that point decided I was going to change my career and focus entirely on malaria, and became the director of the Johns Hopkins Malaria Research Institute, which I am now. And I'm going to just show some slides of the relevance of aquaporins to malaria. Red cells have an aquaglycoporin that lets glycerol in, and the parasite can grow. So an individual red cell infected by one parasite, but it'll grow and divide, and pretty soon we have 32 daughter cells, and it breaks, and this releases a large number of parasites. And the mosquitoes that carry the malaria parasites have aquaporins. When the mosquito bites you and fills its lumen with the blood, it's doing so because the hemoglobin is needed for egg development. Female anopheles will do that, and female mosquitoes, other species will, but they take up so much blood they can't fly very far. So they have aquaporins for fluid management in their Malpighian tubules. Excuse me. So malaria, it used to be a worldwide problem. We had it in the United States until about 1950. Malaria is still a very big problem in the developing world, most notably Africa. And our studies in 3 different countries let us meet many wonderful people. Here I am talking to Chief Macha in southern Zambia. He's the elder who gave us permission to do field studies in rural Zambia. Here's Philip Tuma, a Johns Hopkins pediatrician who grew up in Africa. His father was a medical missionary. And Phil returned to Africa after his training at Johns Hopkins and is taking care of thousands of children in rural Zambia. And of course, malaria is a problem of children. 500,000 of these kids will die every year, and probably several million may survive with permanent brain damage or kidney damage. It was the gift of Michael Bloomberg, former mayor of New York, Johns Hopkins graduate, and I'd like to point out an Eagle Scout who endowed the Johns Hopkins Malaria Research Institute that I direct. So Mike Bloomberg is giving back in a magnificent way. So here I am with a research group in southern Zambia. These are all graduates of universities in Africa. The work has had a tremendous positive effect on the management of malaria in southern Zambia. In the 20 years we've been at it, the prevalence of the malaria has been driven down to about 5% of the original level. It's not down to zero. Our work is not done. It will never be done until all malaria is gone everywhere. But using the scouting skills, our field teams, aided by GPS instruments, they didn't have those back when I was a scout. But there are no road maps in the outback of Africa, and in these remote locations are where the subsistence farmers live, and they're impoverished, and they're glad to have teams come out and screen to see who is carrying malaria, because if the children develop malaria, they can be lethally affected. So I'm going to step through these quickly. These are wonderful people, but they have very simple lives in terms of the technology. They have not yet joined the modern microelectronic age, with the exception of cell phones. Cell phones are prevalent throughout sub-Saharan Africa and allow health monitors to communicate diagnostic statistics minute by minute. The farmers use implements that probably have been discarded in the United States 150 years ago. The children help by bringing the corn to the village to have it ground in a cornmeal, and they know how to have fun. Not one of these kids has a Game Boy, but they're taken care of and educated, and they can live happy lives, and the value of education is changing their lives also. This photo is a favorite of mine. In Zambia and Zimbabwe, where we work, the language of instruction and commerce is English. And if you look carefully, the little girl's notebook says, "School is cool." It's upside down. Working in the Congo, things were more difficult. This is a place of tremendous turmoil for generations. This is the site of Dag Hammarskjöld's plane crash. The Secretary General of the United Nations was killed in 1961, and the conflicts that led to his death are still around. Here's my dear colleague, Bill Moss, professor at Johns Hopkins, after lecturing to Congolese medical students in Kinshasa. And the part of Congo we're working in, Katanga province, is very low and marshy. There are no paved roads. And back in these villages, the children are very vulnerable, and most of these kids will develop malaria at one point or another. And not all of them will survive. It's a pressing need, and we need to pursue this. Finally, in Zimbabwe, things were made even more difficult. There was a dictatorship led by Robert Mugabe, who held power for 37 years and corrupted the country. Malaria went out of control. But we teamed up with a not-for-profit group in Harare to do regional surveillance in the countryside, and that agency is led by Shungu Minati, who turns out to be Mugabe's niece. She's a very dedicated colleague, and we think the world of her, but it shows that sometimes you have to make ethical compromises in order to do the work which you feel is imperative. Zimbabwe is a beautiful country. This is the bridge at Victoria Falls connecting it to Zambia. Here's a tea estate in the Eastern Highlands. And here are the people. They use the rivers for washing, cooking, for bathing. They lead lives which I guess most Americans would consider equivalent to summer camp. And their most precious commodity, of course, is their children. This old gentleman is probably about my age. His dream is that malaria will be eliminated from the valley they're living in, so his children and grandchildren will never have to suffer from it. Because in the rainy season, every morning the medical clinics are filled with young mothers bringing their children in with malaria. So this final photograph shows my dear colleague Sungano Marakura in the dark blue shirt. Sungano was a villager, grew up in a small village in Zimbabwe, but was very good in school and made his way to the University of Zimbabwe and won a scholarship to Oxford, where he earned his doctorate. And rather than joining a lucrative European pharmaceutical company, he did what he always dreamed of doing. It's returning to Africa to knock out malaria. He's now the dean of the life sciences at Africa University in Mutare, Zimbabwe. So the lesson I learned from one of my Chinese postdocs, in Mandarin, the word for crisis is wei ji. Wei meaning a time of danger, ji, a time of opportunity. And I think we're really at a point in world history where the dangers are apparent. COVID pandemic has mushroomed over the last 2 years, and no one would have predicted that. The needs for treatment of drug-resistant tuberculosis, ongoing malaria, as well as the global climate change are tremendous dangers, but there are opportunities for people in scientific careers. And so I hope as people grow up and lead careers, they're able to look back and see that the journey was interesting and the results were useful, but the colleagues that we made and the friendships we made were very rewarding. And it's gratifying to be at the stage I am now. I'm 73, to still be able to call back on scouting experiences as sort of a moral compass and a guide for decisions that needed to be made. So if there's time, I think questions are, I'd welcome your questions.
We
Yeah,
absolutely have some time for questions. So as always, everyone, if you have a question for Dr. Agre, toss it in the chat and we'll ask it. I think I have a question just to start off with. I know you spoke a lot about your time in Scouts and how it impacted you. When you think back, is there sort of a favorite memory that you have of your time in Scouting?
I think the wilderness canoe trips were all of them very special. Of course, any of you that have been to the Northern Tier know how beautiful it is. Of course, it's not always warm and sunny, but I think those experiences were probably the most rewarding to me. And so when my son became a scout, a troop here in Baltimore, and they had a lot of wonderful activities, but they had not gone to a high adventure scout camp for decades. So I was eager to bring the troop to the boundary waters or the Northern Tier. And we also paddled up in Ontario at the Atikokan base and in Manitoba. We went to the Philmont Scout Ranch and the Florida Sea Base. And I think that was something that the boys really loved, and I found it rewarding to be able to do that still.
I mean, it's great that you're able to kind of pass those experiences down to your own family as well. So going to the, your conversation about your time working on malaria, what would you say? I know you put up that chart sort of showing the difference in cases over the years, and there's a really steep drop off between early 2000s down to maybe 2008. What would you say accounted for that really significant change that happened there? Was there a treatment type that came out, or?
Connor, that's an excellent question. And there were a few factors. The discovery of artemisinin. This is a compound extracted from the roots of a small shrub, Artemisia, and for which Tu Youyou in China won the Nobel Prize 2015. The artemisinins are the miracle drug that we've been waiting for, but they need to be used in combination, and they can help treat people who have malaria, but they're not useful to prevent people from getting malaria. You can't treat people with it every day. The introduction of insecticide-treated bed nets, and these are provided from the Global Fund, from the US President's Malaria Initiative, and many church organizations, Lutheran World Relief, for example. That and the aggressive diagnosis and probably some eradication of specific members of the Anopheles genus of mosquitoes have together brought it down. Now, the same methods were employed other places in Zambia and have not had much effect. So it's not a simple process.
Thank you. A question from Robert, also relating to your work on malaria. He wants to know if you believe that it is possible to eventually eliminate malaria worldwide, or at least take it to a point where it is extremely easily treatable.
Well, it is possible, but the predictions that it'll happen in 5 years or 10 years have been all disappointed. It's readily treatable now. We don't have malaria in the United States, with the exception of people who may bring it back from a safari or some tropical vacation where they didn't take the preventive medicines. So malaria is treatable, but sub-Saharan Africa and the places in Asia where malaria persists are the poorest places in the world, and getting access to the bed nets, the medicines, and the organization needed is not there. So we need to do more than just provide medicines. We need to really help these countries straighten out their infrastructure. The corruption, which makes medicines disappear from warehouses and be resold, is a problem. We have corruption in this country too, but it's not affecting the health of others as much as it is in some of the third world places. I think it's possible, but it's only going to happen if this remains a very high priority, because it would not happen otherwise. There were 200 million cases of malaria last year. That's a lot of malaria.
That is certainly a lot of malaria. A question from Joseph. Have you been back to Africa recently? And has there been a notable change in malaria cases in that time?
Well, I was last in Africa 2 years ago, just before COVID. My wife was with me, we were in Tanzania. And I had planned to return to Africa later that spring, but the travel bans made that impossible. People, our program are now returning to Africa, and I expect to do so at the end of the summer. During the COVID pandemic, the treatment of malaria was disrupted because of the fears people had for traveling to clinics. And so there's been a resurgence. It's gone up maybe 20%. So this is going to have to be addressed very seriously. COVID, of course, is killed more than 1 million Americans. And we all are affected every day. I knew this was going to be bad when I went to the restroom at the airport in March 2 years ago, and I saw all the men washing their hands. This is got the world's attention. But in Africa, the death rate is much smaller, probably in part because people are not crowded indoors, and the populations are so young. The median age in Congo is less than age 17, whereas the median age in Japan is 50. So the young people are more resistant.
Thank you. I have a friend who's on, but he's having some trouble with the group chat. So he texted me a question to ask. So he just graduated from Johns Hopkins actually this year, and he is looking to become a physician, become a doctor down the road, go the medical path. And so he's just sort of wondering if you, as a physician and Nobel laureate, if you have any advice for someone fresh out of college who's looking to go down the path of becoming a doctor.
Well, I encourage this person wholeheartedly, and it's a big commitment. Getting into medical school is a challenge. Doing medical school is a much bigger challenge, and then the clinical training afterwards, it's a massive commitment. You start when you're 18 and you don't let up, but the rewards are fantastic. I've talked about my career, and I'm sure I could have made a lot more money doing something else, but I derived tremendous gratification for having had the opportunity to pursue research and then field research in one lifetime. It's been tremendous. It's a wonderful career.
That's great. Question from Colin that I skipped over before. I apologize, Colin. This goes, kind of far back to the beginning of your career. What is it that made you choose to focus on chemistry originally? Was there something as a young person that made you lean into the chemistry field? Yes.
So I showed a picture of my dad in the first part of the talk. He was a chemist. He would bring my brothers and me up to his laboratory at St. Olaf and do little experiments. They weren't experiments, they were demonstrations, but we thought they were experiments. Imagine a colorless beaker, drop with a colorless solution, and drop of what looks like water into it, and it turns brilliant pink in an instant. Drop of another solution, it disappears. I mean, it's the phenomena of science are so fascinating that I found that very interesting, and his dad as a role model. I was encouraged in that direction from an early age, as were my brothers. So I think if I had been born to another family, I might have done another career, but I feel I was very lucky to get that encouragement.
Thank you. That's pretty neat that, so much of your future career was shaped by your father and your upbringing. Robert has another question and asks if you have any thoughts about how we can address the national shortage of medical providers in the country, with a specific emphasis on primary care providers.
That is so important. We import doctors in this country because our capacity is limited because of the cost. The cost of healthcare in the United States is more than double of that anywhere else in the world, but we don't live longer lives. We're doing something wrong, and importing doctors from other countries means that we're taking doctors from Canada and South Africa, and of course they're taking doctors from Zambia and Zimbabwe, and so there's a net flow of doctors from the poorest countries to the richest countries. I mean, this is totally unsustainable and unfair. I think the investment of the public in science and medicine has been tremendous. I think the problem is that politicians have different views on what should be cut. the far right wants to do away with personal injury lawyers. The far left wants to do away with insurance companies. Everybody has a solution that will affect someone else. I think we need to be able to graduate our own. There are plenty of people who are interested in careers in the health sciences, but it's very competitive and it's very expensive. I'm sure my dad would like to have gone to medical school, but he grew up in the Depression and had to work his way through the University of Minnesota selling shoes at 25 cents an hour, and it was not possible. And I'm sure that's true for many prospective health professionals in this country. They don't come from a family that has the resources where they can really focus on their studies.
Thank you. A question from Brian. Brian says, I'm sure that you've observed a lot of change in the biological and medical sciences over the course of your career. For example, he says, now we can get protein structure from cryo-EM. Are there any changes that stand out to you in particular, or are there any new developments in the fields that make you specifically optimistic?
Well, the changes are coming so fast that it's mind-boggling. But when I started medical school in 1970, it's a long time ago, it seems recent to me, our first lecture in biochemistry was by a man named Albert Leninger. He's very famous because he wrote the Leninger textbook. And he welcomed us to Johns Hopkins and he said, I was going to lecture on RNA today, but a paper was published 2 weeks ago in Nature describing an enzyme that translates RNA back to DNA. DNA is sort of the bedrock of memory for biology. And the rule was that it's translated into RNA and that's translated into proteins. But you can do it in reverse. That has led to the revolution in molecular biology that allows us to clone proteins, express proteins, mutagenize them. With the CRISPR-Cas, we could repair mutations. And the discovery was made by two young faculty members at Johns Hopkins, Dan Nathans and Hamilton Smith. And I think that in my lifetime was a revolutionary advance. And there have been many others since then. And things are going to continue to change. The difficulty is as you get older and you get more administrative duties and opportunities to speak or visit, the technology bypasses. So it's makes a career ever so much more challenging. But the improvements in health are dramatic. I mentioned I'm suffering from Parkinson's. This is my 12th year. I would probably be crippled in a wheelchair if it weren't for the medicines that were discovered and prepared that allow us to compensate partially or maybe even fully. And so if you ask anybody 50 or older, do they have any medical problems? Virtually everyone will say yes, they're being treated for high blood pressure or high cholesterol level, anxiety, psoriasis, asthma, and the improvements have all come through medical science.
Great. Well, thank you very much for that answer. I think we have time for just two more questions here. The first is from Cameron. Cameron's the young man who just graduated Hopkins and is looking to go to medical school next year. He says, how difficult was it to connect with the communities that your project in Africa worked directly with? And how did you and your team overcome initial cultural barriers in order to build trust and encourage cooperation?
Cameron, that's an excellent question. Our first work was done in Zambia, in the Macha district of Choma province, southern province. And Philip Tuma, the Johns Hopkins pediatrician, I showed his photograph. Phil has worked in that area for decades, and his father before him. And so they established tremendous trust with the people in the region. And so organizing field scientific groups to enter the homes and draw blood from the children, which is a cultural taboo, it was made possible because of the trust that was established through Dr. Tuma. I think you can imagine if some unannounced medical specialist that was from outside of the district showed up and wanted to draw blood from children, that would just not be possible without the trust that was established. And by dealing with the local leaders, I showed a photo of me talking to Chief Macha. These are not witch doctors. They're tribal leaders are very knowledgeable. Chief Macha is a university graduate, and he insisted that he be convinced that the work would be safe and be undertaken with the highest level of integrity. So that challenge is important, and it's not automatic.
All right, thank you. And just one last question. This is from Robert. What advice would you give to a new Eagle Scout to prepare him or her for adulthood?
Well, I'm not sure I have a question for them, but I would encourage them and point out that we're proud of what they've done, but their work has just begun. To give back to the community from the level of understanding you've lifted yourself as an Eagle Scout is a great reward. One thing I noticed when I joined the faculty at Johns Hopkins, there were Eagle Scouts galore. The dean, Ed Miller, was an Eagle Scout. The chairman of medicine, Ed Benz, was an Eagle Scout. The chairman of biological chemistry, he was an Eagle Scout. It's a pretty nice club to be a member of. So it's written in the New Testament, unto whom much is given, much will be required. And I think Eagle Scouts, we expect much. And they're fully prepared to provide that.
What a great point to end on. Well, I just want to say again, unfortunately we've run out of time, but thank you so much for joining us. This has been a fantastic conversation. I know that you're a busy guy, so we appreciate you taking the time. So everyone give Dr. Agre a virtual little round of applause. And
I'm going to make a real quick
turn it over to Colin, who has a few announcements. So Colin, go ahead.
Just really quickly, thanks again, Peter, for coming on. It's a great presentation, very informative. This week is our council giving week. We have a $50,000 match from one of our generous donors. So any donations that come in this week will count for double. But there's other ways you guys can help too. There's 5 ways that you guys can help during giving week to really make sure this effort is really great. And one is consider making a gift, but 2 is to help spread the word on social media. You can share, tag, and like the posts we've been making. You can share photos or stories on your platform about how scouting has influenced your career, similar to how Peter did during this presentation. And you can visit our Facebook throughout the week. We're going to be sharing stories, tips, and inspirations on how scouting can positively impact workforce development. That is our theme this year, that scouting can prepare future leaders for the workforce. And finally, also, we do have ways to create a legacy with NESA-DC. So a planned gift through endowment is a powerful commitment to our work and mission for years to come. So those are 5 ways that anybody can help during the giving week. We've had a really strong week so far, and any support is really going to be great for scouting. Thanks, Peter, again for coming through and for doing this. And I hope you guys all enjoyed this lunch. We're going to go on a little summer break after this one. So no lunch in July or August, and we will be back in September. So look for the announcement there. Thanks, everybody.
Thanks everyone so much. Thanks again, Dr. Agre, for joining us.
You're totally welcome. Thank you.
Thank you, Colin, for doing this.
Thanks, Bill. Thanks for coming on.
Yep, enjoyed it immensely.
Great. And thanks, Tom. Thanks for coming out, Tom.
Finally, I got out of the classroom, buddy. Thanks.